Hi, this is a kind of a supplement really to the lectures 9 and 10. There isn't enough time during the lecture to cover it, but it is needed information to help you prepare for the lab where you're actually going to do plasmid purification. We call them mini preps because we just grow a small amount of the bacteria and then we lyse them and isolate the DNA. Okay, and so here's the problem. When we isolate, we break open the cells, we have tons of DNA in there, and some of it is bacterial chromosome, and some of it's plasmid DNA, and there's not that much difference in terms of DNA between the two. So what kind of features of the plasmid could we use so that we could make a nice prep that just has your plasmid in it without all the bacterial genes as well? And so there's two different methods. The first one is separation based on size, and we do this a lot in the lab, but we're not going to be doing it in our lab. But it's useful to know how to do it. You basically start, there's a feature that you need to know about, which is the bacterial chromosome is large. And it is often affixed to the inner part of the cell membrane. And so what will happen is you have to break the cell walls pretty gently. You use an enzyme called lysozyme. You have some lysozyme in your tears as a way of protecting your eyes from bacterial infection. There's a bit of EDTA in there. There's a lot of sucrose. So the sucrose is really important. It's a high percentage. And what that means is when the cells break apart, they kind of, the density of the solution keeps the membrane and so on pretty intact at the beginning, okay? And you throw some RNase in there as well. And you end up with what we call spheroplasts. This is a spheroplast. It is a bacterial cell that's mostly intact, but the cell wall has been really disrupted. So it won't take very much now to break that open. So the first step is just kind of making the spheroplast. The second step, you're going to add Triton X100, which is a very mild detergent. It just gently breaks open the cell membranes, and of course the cell wall bits are there too. And what happens is that there are two things that are going on here. The little plasmids are very tiny, and they are also not attached to the cell wall. The bits of bacterial chromosome, they won't be fully intact. They do get a little bit broken, but they tend to break into much larger pieces, and some of them are still attached to parts of the cell wall. Okay, so after doing that, you then do centrifugation. And the centrifugation, all the big pieces of DNA, including all the cell gunk, goes to the bottom. And then in your, what we call the cleared lysate, this is a lysate because the cells are broken open. And cleared means all the junk, the big stuff, has been precipitated, or not precipitated, but spun out. Pelleted at the bottom. And then you basically remove that into a tube, and you would do something like phenol chloroform extraction to make sure there's no proteins, no nucleases, and so on in there. And then we would use alcohol precipitation to remove metabolites, and we're really talking most of the time, most importantly, that should be an A, about salts. And then some of the extraction buffer components, we want to get rid of those. And when we make the DNA in a nice pellet, we can look at the size of the pellet and decide how much buffer to resuspend it in, TE, and that allows us to make sure it's at a good concentration. So that's kind of the size-based method. And remember back to lecture 9 where I talked about conformation. If we have been gentle and careful, most of these plasmids are going to be supercoiled, right? Because they won't have been damaged in the process. What's interesting about handling is that if a piece of DNA is large, your handling is more likely to break it than if the piece is really tiny. the little pieces are slightly protected against sort of shearing forces and stuff when you're working with it. The second method is the one we're actually going to use. It's based on conformation. So it isn't about size, but it is about whether the plasmids are supercoiled or not. Again, we have to use a really gentle technique. And it's this method is most widely used, and we're going to be using a kit, but the kit basically goes through these processes. I worked for many years in a lab with very poor funding, and so we had to make our own solutions and so on, and we still managed to get a good yield from this. So basically, you do cell lysis with SDS. The bacterial chromosome breaks into little pieces, and it gets denatured into single-stranded DNA. We increase the pH, we make it very basic, and the DNA unwinds, and I'm just trying to think the best way to say it. I think coming up I'll say more, but the idea is the bacterial chromosome breaks into fragments and gets denatured. Because the plasmids are supercoiled, they are slightly protected from the denaturation. It's not that it won't happen, but it takes longer because they're all twisted up, and so it's harder for the change in pH to immediately affect them. And hopefully, yeah. And so this is how it works. You basically you do what's called an alkaline lysis. So you basically take your cells, you suspend them, and then you increase the pH, and you do it for a very short period of time, and the DNA all gets disrupted. But notice that the supercoiled plasmids are still pretty intact, whereas the single-stranded pieces are from the big bacterial chromosome. And then you rapidly neutralize. So you do it really fast, and so what happens is the pieces that have been denatured, they don't have sufficient time to find and double strand with the correct complementary pieces anymore, and so they end up just kind of little bits that are complementary will start sticking to each other, but it'll be all the wrong pieces, and so you get this tangled mass of DNA. But the supercoiled plasmids, even if they started to denature a tiny bit, they can just snap back because they're still in that very tight conformation. So then you follow that with centrifugation, and now you're going to have this single-stranded and sort of denatured and clumped bunches of cell wall material and your linear DNA molecules, but you should have your supercoiled plasmids in solution. And we purify them over a column in our prep, so we'll talk more about that when we get to the prep for the lab. But we can also do things like, just as was mentioned previously, you could do phenol chloroform extraction and then alcohol precipitation. In our case, we're using the columns. And if you want to remember how those columns work, if you go way back to the DNA isolation lecture 2, I think it was, that describes how the columns work with the you bind to the column, you wash a few times to wash all the stuff you don't want away, and there's alcohol in there to keep the DNA out of solution stuck to the little beads of the column. And then you dry the column really nicely so that there's no alcohol left, and then you elute in a solution that allows the DNA to come off the beads, and now you have a nice little solution with a whole bunch of your DNA in it. And plasmids are one of the types of DNA that you purify that way. And in large scale genomics facilities like the BISC sequencing center and so on, they have these things pretty much automated. So they have robotics. So they have these gigantic plates, not the little ones like we're going to use, and they've got colonies. I don't know how well you can see it, but every one of these little white bumps in here, these are all individual colonies. And so the robot is just like picking the colonies, and it's also keeping track of exactly where each one is on the plate. And then it goes through machines that take it in, a whole bunch at a time. They're putting, the alkaline solution in there to increase the pH, and then it's timed 5 minutes, and then it's neutralized, and then it's spun in a certain way, and so on. So it basically if you work in the, I had a friend who went and worked in the Genome Sequencing Center, and she was almost in some ways disappointed because what she did was she labeled things and she put things into machines and took them out, but she didn't get to do any of the actual work because the big robotic systems were doing it. But the truth about some scientific processes is that interpreting the data is actually the most fun. So doing it can get repetitive, but you know, at least for her, she got to, you know, look over the sequences and, you know, decide which things to pursue and that sort of thing. Okay, I think hopefully that is a very, very quick run through just the basic principles of the isolation. I used to include it in lecture 9, but as you knew from lecture 9, we didn't have time to finish everything as it was. So I think this separate little video is probably helpful because some of you have already done mini preps and so on, and some of you haven't, and those who haven't might find this useful. Okay, that's it.